welcome to a community of scholars. My name is Aradhana Natarajan, and we're here for part 2, where I talked to Dr. Bingham about his research and discovery of a new cancer drug. So, I'm personally more of a neurology and neuroscience person as opposed to like cancer, but I always have had an interest in genetics. So, as far as I understand it, we have a lot of understanding of the of certain mechanisms that contribute to the development of cancer and the genetic factors and chemical factors that play a role in its development. But how far would you say that we are as far as the progress towards a cure is concerned?
So I think it's fair to say that we know many, many times more about cancer, its etiology, its origins, than we did, say, 10 or 20 years ago. So in one sense, we've made enormous progress. But if our assay is how much progress have we made in improving patient outcome, not as much as you'd like. Patient outcome is still not, hasn't improved nearly as much as we would like. Now, part of the reason for that is that in order to really effectively therapeutically attack any disease, including cancer, it takes essentially complete command. Or you have to understand everything, all the details of the disease that are relevant to therapy. So, for example, HIV, a relatively simple disease because the retroviral life cycle is very well defined. You knew enough to go in and attack it in an intelligent way. We're just now getting to the point where we're starting to complete the picture of cancer enough that we can start to do that. So there have been a couple of major developments in the last several years that result from that increase in command. One of these areas is immunotherapy, for example. We understand the cancer immune response much better than we used to, and we're able to manipulate it now in systematic ways that can produce some dramatic improvement in outcomes. There's still problems. Not all patients respond to immunotherapy. There are problems with autoimmune side effects, but it's a big step and one that we can build on. We have chosen a different route, which is to attack cancer metabolism. We can talk about that if you like.
Yeah, that would be great. So, as far as I understand it, your drug works by targeting enzymes within the mitochondrial functioning of cancer cells.
Correct.
So, for our viewers, the mitochondria is essentially called the powerhouse of the cell. It's what's responsible for taking the carbohydrates you eat and converting them into energy. And enzymes are responsible for taking the chemical structure of the carbohydrate and converting it into what's known as ATP, which powers most of the biological functions within your body. So, how specifically is the mechanism of the drug work?
So let me actually back up one step from mechanism and then walk to mechanism. So cancer develops when the social contract between healthy cells that normally build orderly and well-organized tissues breaks down. And then typically as a result of a genetic mutation, most commonly. And when that social contract breaks down, these cells now start behaving anarchically. That is, they grow in an uncontrolled way, they compete with one another. And they build what is a tissue, a tumor tissue, but one that's, because of their anarchic competition, is disorganized. As a result of that disorganization, the tumor environment tends to be oxygen-starved because the vasculature is not very good. Tends to be nutrient-starved because the vasculature is not very good. You can't get the oxygen in, you can't get the CO2 out, and so on. So the breakdown of social contract produces this exotic environment, and cells have to then evolve secondarily with new genetic changes, with also epigenetic changes, to be able to survive and thrive in this new environment. So at the end of that process, when they become advanced metastatic disease that shows up in the clinic, these cells are highly evolved. Their metabolism has changed to deal with this exotic environment. That changed metabolism, in turn, becomes a set of targets we can shoot at. Remember the problem with shooting at cancer cells. They're your own cells that have just gone off the rails a little bit, right? So it's very hard to hit them without hitting your normal cells. And in fact, a lot of early cancer drugs were quite toxic for that reason, right? Because it's hard to hit cancer cells without hitting your own cells. But this evolved metabolism that we've been talking about here, we and others thought would provide targets that we can hit. Among those targets are the regulation of mitochondrial metabolism. So tumor cell mitochondria have to cope with reduced oxygen, reduced access to nutrient stores, and so they redesign their metabolism. They upregulate certain regulators, they downregulate certain other regulators. They redesign their regulatory systems. that's a target you can shoot at. So we've developed a drug that selectively targets the regulatory systems evolved in cancer cells. Right? And so we can shut down mitochondrial metabolism selectively in cancer cells, and that in turn allows us to have a significant clinical impact.
Okay. So, like you mentioned before, cancer cells are our own cells. They've just changed because of various factors and their genetic makeup and their metabolism, and all of that has shifted. And there's also research that shows that we are constantly producing cancer cells within our body, and that the only reason not everyone immediately develops like giant tumors is because our own immune system is working towards protecting us. And so most of the therapies that are given are immunotherapies, or therapies that target the immune system, or they go the opposite direction, just kill off cells indiscriminately. So how is yours specific? Does it go after the specific genetic makeup of cancer cells? Any chemical indicators that are specific to cancer cells?
So it goes after the evolved genetic and epigenetic changes. So it targets cells based on a new spectrum of regulators and metabolic enzymes that are unique to this evolved adaptation to the tumor environment. And we can get down in the details. It's actually the regulatory kinases that control the pyruvate dehydrogenase enzyme. Let's not go into that level of detail. But we understand how it works. Let me be very clear that even drugs, though, like ours, that are designed to disadvantage and ultimately kill tumor cells, probably work because as you start to kill tumor cells, the tumor cell suppression of the tumor immune response is weakened, and the immune system comes in at the end and cleans up. So, in fact, tumor immunity is probably central to all cancer therapies, whether they are overtly immunotherapy or not. Immunotherapy is designed not to try to kill tumors directly, but to try to hyperactivate the tumor immune response and use the immune response to clear the tumor from the get-go. Probably as we move forward, some combinations of these things, where you disadvantage tumor cells and you activate the immune system, the tumor immune system, for example, will be the way that it plays out. You're right also that you and I have new potential tumor cells arising in our bodies every day, even a healthy young adult like you. But normally a set of processes, including tumor immunity, protect us from that. Also, our cells are designed so that if they take the first step toward malignant transformation, they tend to be killed. They tend to be instructed to undergo cell death. So natural selection's put a whole bunch of barriers in place to allow us to be long-lived creatures without perishing from cancer. Tumor immunity, though, is one of them. It's a big, important one.
So, since you've discovered the drug, right, how far has the development stage gone?
So we've been very fortunate. We were basic researchers who didn't really set out to do clinical research, but we came upon an approach, and then we were approached by people in the pharmaceutical industry to license the patent that we had and support this process. So we discovered the drug, they developed the drug, and we're now quite far along. So the drug has been through what are called, in the jargon of the area, phase 1 and phase 2 clinical trials. These are investigational trials to try to get a feeling for whether the drug works. Those trials are published, so I can talk about them. We got a very nice signal in those initial trials. The signal was strong enough and promising enough. This is now in the area of pancreatic cancer, called PDAC for short, pancreatic cancer, which is an extremely difficult cancer to treat. People who present with stage 4 cancer, very advanced cancer, metastatic cancer, have a very grim prognosis. And so our drug significantly in investigational trials improved that prognosis. We added significantly to survival and the number of patients who had complete remissions or complete responses in the investigational trial. So that's hopeful. So now we're going into what's called phase 3, where we have a larger trial, multiple centers, to make sure that any success we have is robust and reproducible, and with large control sets and so on, so that we can be very confident. If we see the same effects, be very confident. We're hopeful that that trial will be successful. For those of us here at Stony Brook, the recent news is that one of the centers engaged in this trial is in fact right across the street over here. This is the Stony Brook Cancer Center. Professor Min-Sik Choi is in fact the PI, the physician directing that. And so we're looking forward with particular interest in the patients that will be treated here at Stony Brook with this approach. And again, it's too early to know, but if this phase 3 trial is successful, then this drug can become part of the standard armamentarium against pancreatic cancer now and other cancers in the future.
So that's actually really impressive. That's pretty cool. So, but you have always been more of an academic researcher. So, what has it been like entering medicine and working with medical institutions?
So I'm a basic researcher by training. I'm a PhD, not an MD. And so, you normally expect as a basic researcher that you hope that things you discover will be used by others in the future to improve human welfare. But we have had an exceptional good fortune to be much more close to the human welfare connection. That is, patients are now actually using the drug that we've originally discovered. It's been, first of all, enormously emotionally rewarding. I've met some of the patients whose lives have been saved by the drug. As a non-physician who doesn't have these kinds of experiences all the time, it's a really gratifying experience, very emotional. I feel great gratitude at having had this experience. But what I've also learned is what a complicated and challenging process it is to get from the first stage of discovery. You have something you think might be a drug, the number of steps and the huge costs and effort to get to the point where a patient walks into a doctor's office and this drug is prescribed. I had no idea how many moving parts there were to that process. So you need risk-taking investors, you need the regulatory agencies, there are a whole set of development specialists who figure out how to formulate the drug for human use and so on. It is a huge process. And of course, back when we discovered the drug, I didn't know any of that. And so it's been also an educational journey for me to see what it takes to get this done. It's impressive how many people have to be involved.
Okay, yeah, that sounds really cool. And I wish you the best of luck going forward.
Thank you.
And thank you so much for being here.
It's been a pleasure.
And that wraps up our session for the Community of Scholars. Thank you for tuning in, and we'll see you later with more content.